Welcome back to the course Fundamentals of General Immunology. Today is our lecture number 11 and the title is Intracellular Antigen Routing and Processing Pathways. So in this lecture we will try to understand how antigens are processed, how antigens are the uptake of antigen by the cellular system, and how these are processed through the pathways which exist in the cell population. We will also try to understand what are the different ways these antigens are processed, and based on the processing we could expect the different kind of immune response generated upon processing of these antigens. So when we try to understand the general pathway of antigen entering into the cellular system, there could be multiple pathways. Just for example, if we talk about the respiratory pathway, so for example, if we just try to understand, this is just for general understanding, that if there is a small intranasal delivery of any biomolecules that's happening through the nostril, this can goes through the nostril and the epithelial area of the nostril, and in this area we have these trigeminal nerves, and through these trigeminal nerves this get internalized upstream towards the brainstem or towards the mucus layer. So let's try to see that what are the things. So suppose these are the cerebrospinal fluid which are part of the central nervous system, and then we have a mucus layer which is a part of the outside of the respiratory epithelium, and of course we have the ciliated respiratory epithelium that allow the beating of the cilia in either direction, either in the forward or in the reverse direction. So these biomolecules which are present in the mucus layer, which are coming from the spray of any any system, then these biomolecules can be uptake towards the different areas through these trigeminal nerves. So there could be a possibility that these trigeminal nerves can breeze through this cerebriform plates, and there could be a possibility that these trigeminal nerve can push the material or push the biomolecules towards the brainstem or into the cerebrospinal area. So there could be a different mechanism altogether when we are talking about nerve based entry of the antigen or biomolecules. Of course, when we are talking about biomolecules, antigen is also considered as one such biomolecule. It could be a protein, it could be any other substances such as polysaccharide and all. So let's try to see a different example and say there could be a possibility that olfaction or olfactory route may also possibly involved in the trafficking of those antigen from the nostril or the nasal epithelium. So again the same thing, suppose if there is a spray of the biomolecules to the nostril area, we have the nostril epithelium, and of course we have this olfactory bulb that could be giving an entry of the antigen from the mucus layer. Of course we are talking about this biomolecule. Now try to understand here, there could be three possible ways the antigen could enter into the central nervous system. So of course this is the fluid which is a part of central nervous system called cerebrospinal fluid, and of course these are again olfactory epithelium, and these are the nerves which are giving in the olfaction called olfactory nerve, and the possibility of these antigen to enter into the system could be possible through three different ways. First could be called transcellular pathway, where the molecules are entering through the cellular system and going to the upper area. The second could be a paracellular pathway. Paracellular pathway stands for when we have an in between epithelial. So if the marginal epithelial layer in between the two cells can give an entry of antigen to the cerebrospinal fluid or into the central nervous system. So this could be the second way, and third could be through this nerve called intracellular, which is kind of in between two cell types. So basically it is through the cellular system. Intracellular stands for inside the cell. So these are passing through the neurons, olfactory nerve to the olfactory bulb. So basically, there could be a possibility that the antigens can enter through the cellular channel called transcellular pathway, or through the lining in between the two cells, or there could be a possibility that it could pass through this nerve cell directly into the olfactory area. Now, these are the ways antigens are processed. So, cellular systems are very complex, and therefore, we could see that there are varieties of ways the antigen can be taken by the cellular system. Now, having this in mind, the cells are also very complex. So, we are talking this in a holistic way when we are trying to see what is happening in a general way, that once the antigen is entering inside the body system, how these are taking care by the system which are available around those entry point. So, this was an example of nostril. The similar thing exists when the things are going through the gastrointestinal cavity, say from the mouth to the gut. Also, there could be a possibility that the antigen can be processed through the eye, where the lacrimal fluid and the other things present in the eye ball area could modulate the entry of the antigens. So, the cellular entry of antigen depends upon the type, cell types, and also the metabolic aspect of the cellular system. So, we could look into that in more detail in the coming slides. So, this is just a generic overview of any endocytic pathway. When we call endocytic pathway, it stands for the pathway which exists inside the cell, which is present inside the cell. So, these are all the extracellular matrix. So, these are the proteins which are present on the extracellular matrix. These are also helping the cell to cell communication, cell to cell homeostatic balance. This could be the channel protein which are present on the cellular surface. This could be involved in active process or in the passive process, and these can give an entry to the ions or give a passive or active entry to any other biomolecules. Now, for example, when the biomolecules are entering inside the cellular system, there could be a possibility that they can engulf through the phagocytosis or pinocytosis. It all depends upon what kind of antigens. If the antigens are in the particulate form, we called it phagocytosis, the cellular eating, phagocytosis. And if the antigens are in the soluble form or in the very they are not in the particulate form, then we call it the pinocytosis, which is a cellular drinking. So, the process changed based on the type of antigen. So, this could be a particulate antigen called phagocytosis, or it could be a soluble antigen which is called the pinocytosis. By these two processes, the antigens can enter inside the cellular system. Now, once they are entering inside, this cell membrane invagination can make a small kind of early endosome like structure, and because of the acidic environment, we could see the processing of the antigen inside the early endosome in a different way. This could mature in a different form, and one such maturation is increase in the pH. Say H plus ion can be internalized to increase the acidic environment of the endosome, and this could lead to the conversion of early endosome into late endosome or the mature form of the endosome. Then this small kind of a pinched out antigens can be gone or can be processed through the Golgi apparatus, and then it could come out through the exocytosis in the form of a vesicle. Now, this could be a pathway which can regulate the cellular homeostasis. Now, what is happening here, that bigger molecules are taken into the cellular system, and then through the processing through Golgi apparatus or endoplasmic reticulum, these are processed through the smaller one or the smaller particle, and then they are exocytosed to the cellular system or extracellular matrix by this exocytosis pathway. So, this could be an endocytic mechanism where the antigens can be internalized through the endosomal pathway, and through this channel of Golgi apparatus or endoplasmic reticulum, it can be processed in a different way or variety of different ways, and then go out to make its functionality. Now, these all processes are very much synchronized, and these are needed in order to make a functional antigen. So, the processing of these proteins are important in the cellular system. This could be done in order to facilitate the cellular homeostatic mechanism in terms of nutrition, in terms of metabolism, and also in terms of processing of the foreign antigen. We will try to focus our discussion here on the processing of foreign substances, because that's the theme of immunology, that how these foreign materials are processed through these pathways. Let's try to see a generalized picture what we have discussed in an endocytic pathway, that once the things are endocytosed, we can get a vesicle, and then this vesicle could form an endosome like a structure, and this can radially go into a different cycle, and there could be a process called autophagosome formation or a lysosome formation or an endosome. So, basically once the things are internalized, they go through the various channels in order to do a functionality, and accordingly the processing of the antigens are done, and so that they can be making they can make a homeostatic environment throughout the cellular system. So, just to take home message through these two slides which we have discussed about the generic endocytic pathway is, antigens are entered, and after entry, based on the antigen biology or based on the requirement of the antigen, they can go through the endocytic pathway where the endosome is involved. They could go in a maturation in a different ways to endoplasmic reticulum or Golgi apparatus, or they may go to lysosomal structure to get a different pathway altogether. And sometime there could be a auto phenomena in the cellular system when there is no nutrition. We could see that there could be an formation of autophagosome that could lead to autophagy kind of process in the cellular system. So, there are multiple ways the cells can be maintaining the homeostasis. Let's try to move on, and now we will try to see that how these pathways are defined. But before that, let's try to see that what are the different types of antigen presenting cell. Now, when we say that the antigen is processed through the cellular system, we also looked into how these antigens are uptake through the neuronal pathway or through the olfactory bulb in the earlier stages when the things are entered through the nostril. Now, there are three main types of cell. There are many other types of cell present in the human body, but there are three main types of cell which are called antigen presenting cell, which stands for they can present the antigen to the further processing. And there are three types which are mostly identified. One is called dendritic cell. The name itself called dendritic cell because there is a dendrites. These are the projections which are coming out from the cellular system, and this can facilitate the higher surface area in order to trap the pathogens such as viruses or bacteria or any kind of particulate antigen. There could be a second type of cell called macrophages, which are a bigger cell population. They can engulf the bacteria by a process of phagocytosis, and then we could see that they can start doing the phenomena of presenting it to body immune system. And finally, the B cell is also, the B lymphocyte can also present sometime the antigen to the other immune cells such as microbial toxins, and this toxin can be processed through this CDR region of immunoglobulins, and they can present it to other neighboring cells in order to facilitate the immune response. Now, in human bodies, when we are defining the cell types, we looked into the earlier classes that each cell type expresses a protein on its surface, and each protein expression in a cell type can define what kind of cell it is. And these proteins collectively called it cluster of differentiation, the cell mass which expresses this protein. So, the markers, so this could be a specific marker for that particular cell type. For example, dendritic cell population can be identified by the expression of CD11C. This is a specific protein which are expressed on its surface. So, CD11C is a very specific generalized marker for dendritic cell. While we have, once the antigens are processed, the dendritic cell become mature, and the mature form of dendritic cell can express other kind of proteins such as CD83, CD80 or 86. So, these are the marker for mature dendritic cell. What it implies that the presence of these proteins on the surface of dendritic cell can give you an indication that these are the actually dendritic cell. Well, all the other cell type may not express this protein or may express the protein in a very light quantity or very small quantity. They can occasionally be presented on some other cell type as well, but most of the time these are a specific marker that can express by a specific cell type. Similarly, macrophages can be identified by the expression of CD11B molecule on their surface. That means the macrophages can express these molecules very categorically on macrophages. So, we can identify if we have some mechanism where we can look into the CD11B expression on the cell type, we can say that these are macrophages. Similarly, CD11C can give an indication that these are dendritic cell population. Similarly, the B cells are identified by a universal marker called CD19. So, CD19 is a marker for B cell population. Now, having said that, these are not a very fixed criteria. The cell type, based on the cell type, based on the expression biology. I'll just quote an example here, the macrophages. The macrophages could be of two different types. One is called M1 macrophages or the other is called M2 macrophages. M1 is functionally different and M2 function differently in the cellular system. So, M1 can be pro-inflammatory, it can enhance the inflammation. So, this could be a prior inflammatory response. So, M1 macrophages are expressing a different kind of protein on their surfaces, while the M2 macrophages are anti-inflammatory. So, they can respond against the inflammation. The first one is pro-inflammatory and M2 is anti-inflammatory. So, the expression of the cell type, the protein on the cell type may differ. So, although they are both are macrophages, there are two different types of macrophages and their functions are also very different, and the biology of these two cells can be identified only if we can segregate these two cell population. To segregate, we need to understand what are the markers present on the cell surface. So, there could be many markers which are present on the cellular system, and these are just some examples of these proteins which are expressed on the cell surface. So, these are three different antigen presenting cell: dendritic cell, macrophages and B cell. There are other cells as well. So, we will move on now to the very specifically the processing of antigen through the intracellular pathway or through the cells which can intake the antigens. Now, there are two pathways which are listed, mostly the endogenous and exogenous pathway. Now, let us try to understand the two pathway in a gross way, and then we will go in detail of each cell pathway in different cell type and how they are regulated. So, as name itself indicate, endogenous stands for internal and exogenous stands for external. So, suppose this is an antigen and this is a cell. So, this cell type, and let us say this is nucleus. These cells are right now present outside. So, when they are engulfed by the cell through the process of phagocytosis, these are called exogenous antigen. And if the antigens are processed and manufactured inside the cell, we call it endogenous antigen, right. So, there are two classical ways the antigens are defined. Either they are coming from outside, exogenous, or they are produced inside, called endogenous. Endogenous antigens are mostly coming from viruses, and exogenous antigens are mostly coming from bacteria. So, basically these are not limited to the two different types, but mostly they are defined by their origin into the and how they are processed into the system. Now, once these are endogenous, they are decorated on the cell surface by a classical molecules, and these are specifically called as type 1 major histocompatibility complex, which we will see little later, and the exogenous antigens are processed through this torch-like structure, which is called type or class 2 major histocompatibility complexes. So, remember, there are two ways the antigens are entered into the cell, such as endogenous and exogenous, and there are two different ways these are presented to the immune cell. The endogenous antigens are presented by MHC1 pathway, and exogenous antigens are presented through MHC2 pathway. So, these are two distinct pathways, and we will go one by one. But before going to the types of endogenous and exogenous pathway, or MHC1 or MHC2 pathways, let us try to understand this critical molecule called major histocompatibility complexes. These are again very important protein which are present in the cellular types. Now, what are the different types of MHC? What are the cells which expresses this MHC? Let us try to understand that first. So, major histocompatibility complexes are the surface protein in a cell. So, these are protein present on the surface of a cell, and mostly when we are talking about a protein which are present on the surface of a cell, these are mostly glycosylated protein. So, these are called glycoprotein. That means once the protein are formed into say the functional unit, just to make them the functional protein, the proteins are loaded with glucose or sugar coated molecules. So, the proteins which are produced from a ribosome, when decorated with glucose or sugar like molecules, they are called glycosylated proteins, and most of the glycosylated proteins will come eventually on the surface, and therefore, these are surface glycoprotein. They can bind to the different, or they can present the antigen on their surface. So, we will see it later, but their function is actually they are like a torch like molecules. So, once they encounter the antigen inside the cell, they can present this antigen to the other immune cells like a torch, so that the cells can be sensitized and can start producing an immune response. Second is they are very crucial for adaptive immune response, because these are not the innate immune system. Once the antigens are entering inside the body, they are processed through the MHC1 and 2 pathways, and then based on that you see a different immune response altogether. In human body, these proteins are manufactured or produced by expression of certain genes, and these genes are called human leukocyte antigen genes, HLA in short, and therefore MHC genes are also called as HLA gene. These are located in humans in chromosome number 6. So, the HLA genes are located in chromosome number 6, which can code for these MHC molecules. They are very dynamic molecules, again like antibodies. We have discussed the VDJ recombination. Here also a lot of recombination is happening in the MHC molecule, and therefore these are polymorphic, these are highly polymorphic in nature. They can process a variety of pathogen for recognition through the antibody by different immune cells. There are 3 different types of MHC. We called it class of MHC. Class 1 is especially expressed by all nucleated cell. So, every nucleated cell, virtually all nucleated cell can express class 1 MHC molecule. Class 2 is categorically produced mostly by dendritic cell, macrophages or B cell, particularly the antigen presenting cell, which we have discussed in earlier slides, that these MHC2 are expressed by those cell types. While class 3 is actually not producing any immunity based response or not presenting an antigen, but they are responsible for production of cytokines such as tumor necrosis factor, TNF, or different complement protein, which we have discussed in our earlier classes, that these are the protein responsible for complement fixation, or the processes which we have discussed in earlier classes, such as opsonization or any other complement pathways. So, these are not expressing, although they are not producing any presenting molecule, but they are producing cytokines in the cell. And of course, they can differentiate self from non-self, and they also play a key role in apart from defense response, they also play a very important role on transplant rejection, and also the immunity response against our own antigen, self antigen, called autoimmunity. So, we will see those autoimmunity and transplant rejection in our future classes, but these are some of the functions of major histocompatibility complex. Let us try to see it in a human chromosome, how these genes are located on a chromosome. We discussed that these are located in chromosome number 6, and there are the structure in the chromosome. Let us try to recapitulate the structure. So, the ends are called telomere, which are on the ends of the chromosome. The middle portion is called centromere. The smaller arm is called P arm, which stands for petit, right. So, these are called P arm, and just opposite to P is a Q arm. So, these are the structure, typical structure of a chromosome. And then let us try to see the structure and the location of the genes which can code for these MHC 1, 2 and 3. So, this orange color can code for MHC 1, and we could see that there could be 3 types of human leukocyte antigen, A, B and C. So, these are the 3 MHC 1 types. MHC 2 could be DR, DQ and DP. So, these are the different chains which are present on the HLA. So, either this could be DR, DQ or DP, and particularly the MHC 3 can code for these cytokines called TNF alpha. So, these are the location of genes in chromosome number 6, and they can code for human leukocyte antigen, which we are trying to understand in this whole ways of antigen presentation. Now, let us move on and try to see the typical types of MHC 1 and 2. We will not discuss type 3 because this is just a cytokine, and these are the protein which can do a functionality assay, or this could involve in different function when they go and modulate different cell types. So, TNF alpha we can look into some other classes, but we already have discussed complement proteins when we looked into how these are doing some activities when they are coming in contact with a microbial surface. So, let us try to understand this first molecule, MHC 1, class 1 MHC molecule. Now, if you can look carefully, there are 2 chains here. One is called alpha chain here, and this is actually cartoons showing the 3 domains of that chain, alpha 1, alpha 2 and alpha 3, and these are straight chain, and these domains are actually making a groove here in between the alpha 1 and alpha 2 that can give a docking area to the peptides. So, these are the areas where antigens are presented so that this can inform the other cell type. Of course, these are transmembrane domain which are buried inside the cell membrane of a cell. So, MHC 1, as we discussed, that these are expressed by all nucleated cell. Any cell which can express the nucleus will have the MHC 1 into their surface. So, this is the longer chain, alpha 1, alpha 2 and alpha 3. Remember one point, that antigen, the peptides are docked in between the alpha 1 and alpha 2 region of MHC 1, and there is a small unit called beta 2 microglobulin, which is another segment which are non-covalently attached with the alpha chain. So, there is a non-covalent interaction which are there in between the alpha and beta 2 microglobulin. So, remember the alpha 1, alpha 2, alpha 3 and beta 2 microglobulins are the part of MHC 1 molecules, and the docking area, the peptide binding groove is present in between alpha 1 and alpha 2. This is just a pictorial representation of the same slide in the top view. So, these are alpha 1 and alpha 2, and we can look into the peptides are presented to the other immune system between the alpha 1 and alpha 2. Alpha 3 is just there which can pass through the transmembrane domain, and beta 2 microglobulin is present as a non-covalent entity in the MHC 1 molecule. So, these are a typical structure of the MHC 1 molecule. There is structure resolved on a X-ray diffraction, showing this crystal structure that the protein look like that. There are a lot of mutational study that happened where people have shown that partial mutation in this domain can abort the binding of this antigen to this site. So people have jotted it down clearly that what are the amino acid which can give an entry to the antigen in this groove of alpha 1 and alpha 2. So let's try to move on and see that in a very simplified way. So this is the same thing which we have discussed. So this is a peptide binding cleft here, alpha 1, alpha 2 and alpha 3. This is a transmembrane domain, and beta 2 microglobulin is present here. We have looked into the gene location when we discussed that HLA type where MHC1 is expressed. There are 3 types, A, B and C. So there could be HLA type A, HLA type B and HLA type C. These are very, very important when we do grafting. When the tissue or the cell from one individual is transplanted, or there is a tissue transfer of from one individual to other individual, these HLA type play a very important role. So if there is a matching of the HLA between the two individual donor or recipient, then we don't see any rejection. And if there is a mismatch in the HLA type, say if HLA1 person is giving to HLA-B or HLA-C, then we could see some sort of rejection or the immune response, because these are not coming as a same individual. These are not considered as the same kind of a protein, and therefore the body can raise an immune response. So the grafting, when we looked into the transplant immunology in our later classes, when we study later classes the transplant immunology, then we can understand how this rejection and transplant biology is modulated through the expression of HLA A, B and C types. Again, from the top view, these are the peptide binding cleft, and these are the two chains of alpha 1 and alpha 2. Just to note here a small point, that the peptide size in MHC1 molecule are peptide binding cleft in the MHC1 molecules are smaller as compared to MHC2 molecules. So MHC1 can dock a smaller peptide as compared to MHC2, where in MHC2 we can see that their grooves are wider, and therefore we see a bigger size of the antigen can be coded or can be docked into that site. So let's try to see the MHC2 molecule. Since we now understood MHC1, let's try to see MHC2. MHC2 also the similar kind of thing. Now here the variation is we don't have beta 2 microglobulins. So there are two chains, one is called alpha and the other is called beta, and this is again the variable loop of alpha, variable loop of beta. There is a constant loop of beta and a constant loop of alpha. So this is an alpha chain divided into variable and constant region. This is a beta chain which is divided into variable and constant region. So these are the two specific regions where you see variation, and of course is similar like antibody, that we have variation, and therefore this particular area give an access to an antigen. So this particular area can give an access to antigen. So that means the peptide binding regions are in between the alpha and beta chains here, and this is category called in between the variable chain or variable loop of alpha and beta. So these are the areas where the peptide can be docked. So and then of course the rest molecule looks very similar like MHC1. So there is a transmembrane domain here which can pass through the membrane, because these are rich in hydrophobic amino acids, and of course these are joined by disulfide linkages in order to make a secondary structure and make a complex structure of the protein. So let's try to see the same thing in a different diagram. So this is the same molecule MHC2. So this is actually a beta chain and this is alpha. We called it variable alpha and variable beta, but we can also call it alpha 1, alpha 2, beta 1, beta 2. So this alpha 1 and alpha beta 1 is actually the site where the antigen can bind categorically. So alpha 1 and beta 1 is the domain where we can see the peptide binding. Now if you can compare, the sites are pretty free, and therefore they can give access to a wider peptide length as compared to MHC1 molecule, and therefore MHC2 can give a large size of the peptide binding as compared to MHC1. So what we have understood is how these antigens are processed through these two pathways. One is called endocytic pathway and exocytic pathway, and these two pathways are linked to each other in a different way. One can process through the MHC1, which is endogenous antigen. The endogenous antigens are presented through MHC1 molecules, while exogenous antigens are present through MHC2 molecules. Then we looked into the different types of MHC molecules which are present in chromosome number 6. The gene which can code for the protein synthesis is present in chromosome number 6. There are 3 types of MHC, 1, 2 and 3, and the structure of MHC1 is different as compared to MHC2. So in MHC1 we have alpha 1, 2, 3 and beta 2 microglobulin, while in case of MHC2 we have alpha and beta chains separately. There is no beta 2 microglobulin in MHC2. The peptide binding groups are pretty separate in these two molecules, and also the MHC1 can give a smaller peptide docking, while MHC2 can give a bigger peptide docking in the region. In the next class we will try to understand the endocytic pathway or MHC1 pathway, and then exogenous pathway, MHC2 pathway in details, and how these pathways are critically responsible for presenting the antigen for further processing. Thank you.